record பண்ணுவேன்.
Professor Chin, can I record?
Fine.
Hey, Professor, you going on vacation in a week?
Not exactly.
Nah? You got to prepare for the fall?
Yeah.
You don't give yourself a breather, huh?
Not that much.
Professor Chen, for G, is it incorrect if we put pituitary gland or pituitary body?
Gee. That's fine.
Okay, thank you.
Professor, you live in San Francisco or you live
Pacifica.
Pacifica? What you got a favorite restaurant down there? Where you like eating?
I don't eat out that much.
You like cooking then?
No, I'm a vegan.
Vegan. That's pretty sweet. I'm actually cooking for a couple right now, but they can do vegetarian food. It's been a lot of fun actually. Different perspective.
Okay. Then Just so you know, something about the nerves issuing out of the telencephalon. With some of the lower vertebrae, especially with sharks, the most anterior nerve issuing out of the telencephalon is the one that would be moving to the anterior portion of the head, allows them to sense whatever vibration that would be in the water, so they can easily figure where food is, and where predators are, etc.
Are you talking about humans or like sharks?
Well, on the shark, you can actually see it very large item. We have it, except it doesn't show up readily. So it wasn't until decades ago when they actually found it. And it's a fine item that goes to our foreheads also. It's said to pick up any change in around the forehead. And it's also mostly sensational. And because at that point, all these other ones already numbered from 1 to 12, they couldn't number that one anymore. So they just refer to that as cranial nerve 0. And because all the rest are used with the Roman numerals, ancient Romans, they didn't have they didn't have any understanding about 0. So there's no 0 in Roman numerals. So after naming that, they just refer to that one as a terminal nerve. The only people who knows about this are the ones that had taken comparative anatomy. When I took comparative anatomy, we dissected sharks.
Professor, is 14
Oh.
true or false?
14A is false.
For a question like that, are you looking for upper right of the body? Are you looking for like something more specific?
It's the right upper quarter of the body, not the right half.
Thank you. Could you go back up a little bit, please? Okay, thanks.
How many of these can we get wrong again?
What did I do? Did I put 4 points on this? So if you miss
I believe.
one,
I think you put like 10 points.
yeah, you just have to thank to some of the people in the past couple of semesters. That's when I was giving them 10 points on these. Some people were finding them on the web. So thanks to those people.
For I have a question about 13B, the precapillary sphincters. You don't have to scroll all the way back up, but why can't they prevent blood from flowing into the thoroughfare channel? Don't they have valves? Or it automatically has to go? It has to pass through the thoroughfare channel to get to the venule to the venules. Is that what's happening? Just to clarify, kind of.
The the precapillary sphincters are there to prevent the blood from moving into the true capillaries. Okay. That's all those sphincters can do.
Instructor Jack, could you scroll it down so we can see 17A as well, please? Thank you.
What is the most inferior spinal nerve?
It'll be the coccygeal. Okay. Okay, for the CSF, choroid plexus has to do with those front shape items, has to do with whenever those capillaries pushed into the ventricles, they would then be picked up on the ependymal cells. So with those together, that's what choroid plexus is. And I'm not sure your textbook actually has all these things right, but in most parts of the body, when you need to push blood vessels into something, usually it's sent as close together as possible. So the set of choroid plexus that appeared on the floor of the lateral ventricles, and it's essentially it's the same set of the ones that pushed in here. So you also get the choroid plexus of the third ventricle sitting on the ceiling. Okay. Then the fourth ventricle, somewhat near the bottom. Okay. So the capillaries that pushed in here are the ones that are permeable. So fluid in the capillaries would constantly be pushed in here. So with all of these, they all set up with tight, very relatively tight junctions, those ependymal cells. And they also have ion pumps. So whatever they send in and taken back in, would be active transport. So the type of material that would be going in and out would have to do with oxygen, glucose, vitamins, etc. is what is being sent into the ventricles. And it can also internalize some of the waste material. And when you're looking at the ventricles, so these two are the lateral ventricles, and well, this is way too big, but this item is supposed to be the interthalamic adhesion. Okay, but it's not that big. Okay, so that would be the third ventricle there, and you have a pair of the interventricular foramina that is going to be draining the fluid from the lateral ventricles into the third ventricle. And then from the third ventricle, you would have the cerebral aqueduct that is going to be sending the fluid down to the fourth ventricle. All four ventricles would have fluid going in. Okay, that's the reason with all those black arrows there. Once the fluid gets to the fourth ventricle, then there are three openings there that would allow the fluid to go out into the subarachnoid space. That's a space between the arachnoid mater and the pia mater. So the three median aperture is a single one. Lateral apertures, you have a pair of those. And it's a very, very minimum amount of material that would enter into the central canal of the spinal cord. And because the central canal of the spinal cord is so small, most likely the fluid that had gone in there got absorbed. Okay. So for the remaining flow of the fluid, whatever gets out of the fourth ventricle, the fluid that had gone down in the current, they would be going down in the back and then swing around the spinal cord to the front and then continue back up. And eventually that would reach those arachnoid villi that is going to take that fluid back out into several of those sinuses. So that fluid would now be coming back into the blood. And also back here, any fluid that went into the current that is moving up, it would be swinging up and also continuing to where you have arachnoid villi. There are many arachnoid villi up there, not just these two that is shown here. Okay. That should be at least anywhere between 30 to 60 of them. Some people would have relatively large ones. Some others would have much smaller ones, and those could be probably over 100. So that's a general flow of the CSF. In the adult, you need to have 150 milliliters of that to be replaced every 3 to 4 hours. Over the course of a day, there'll be somewhere around a liter that I've gone through. Okay. So the brain weighs somewhere between, somewhere around 3 pounds. And the cerebrospinal fluid would almost totally reduce that weight simply by keeping it buoyed. And that would reduce at least 97% of the weight, so you don't feel that weight. So again, when people have injury around here, items don't need to go into the brain to kill them. Once the meninges are ripped and the fluid leaks out, once the brain starts settling down, that weight on all the impulses is enough to kill the person. Okay. I think usually when they see large items here at the arachnoid villi, they tend to refer to those as the arachnoid villi. And there would be people with very small items and it's granular. That's when those should be referred to as arachnoid granulations. Continuing into the spinal cord. once the once a neural tube is formed in the embryo, it would eventually have the length of that body at that time. And then neurons and the nerves would build up much slower than everything around. So anything that is being built up by the mesoderm is going to expand a lot quicker than that spinal cord. So when the fetus finally is born, the spinal cord is still almost taking over the length of the torso of that individual, but very quickly it's going to continually the rest of the body is going to be continually lengthening much quicker than the spinal cord. So the spinal cord simply give up on extending, and it would be sending out a whole series of these spinal nerves, literally chasing after all the elongating structures, especially the ones down in the lower limb and the lower part of the torso. That is the reason once you reach adulthood, the spinal cord would actually be ending at the level of L1. By now you know L1, where L1 is on you, okay? So if you go from the part of the neck down to about L1, that's about your length when you were born, okay? So that is also the reason why it takes us mostly more than a year to figure out how to deal with many of the activities in the pelvic cavity. When to urinate, how to defecate without figuring how to block the reflexes. So until all these spinal nerves finally fill and enter and set up with all the material, that's when you finally can control urination, defecation, etc. Also stand up, walk. Okay. That's the reason for it. So at the end of the spinal cord, that item is referred as the conus medullaris. And then from the conus medullaris, there is this item that is referred as the filum terminale. The filum terminale is non-nervous. It's not a nervous item. At the conus medullaris is where it's mostly pia mater, and then a little bit beyond that, it will be the mixture of pia mater and arachnoid mater, and then just a little bit further, it will be totally taken up also by the meningeal dura, and all that is going to continue to the coccyx, and it will be anchored to the coccyx. And then the rest of it are the spinal nerves, because they look like the horse tail. That's why it's referred as the cauda equina. Okay. And a few other things that happens here. Only the meningeal dura would continue around the spinal cord. The periosteal layer of the dura mater ends at the foramen magnum. So what you are seeing here is, this is how spinal cord is accessed in the lab. It's from the back of the person. So mostly they need to cut and destroy all the posterior portions of the vertebrae. Once those are taken away, then you see the spinal cord. Takes a lot of time to do that. And then, so you're looking at the backside. So these items are the posterior rootlets. Your book would refer to those as dorsal rootlets. Once they all come together into one piece, then it would be the dorsal root or posterior root. And this one is anterior root. Okay. And this is where they come together. And from this point on out, spinal nerve. So what you see right here on the surface, and you can't see it, but anytime you see something that looks like it's totally bare, just figure that pia mater is on it. Okay, pia mater is right, is there. It's microscopic. And then there would also be attachment to the dura mater. Okay, so the piece out here is the meningeal dura. And then you also have a mixture of arachnoid mater and pia mater that would attach to it pretty much at every level of that spinal cord. And because they are attached here in the lab, well, this part isn't attached, but in the lab, at this spot, that spot, and something about around there, once you pull on the meningeal dura, then this is going to look like it'll be pulled out and it'll look something triangular. That's the reason it's referred as denticulate ligament. Looks like a sharp tooth. Okay. And the denticulate ligament is the one that make very certain that your spinal cord with CSF around it is not going to be swinging around, bouncing anywhere. It's fully set. In the center of the vertebral canal. So for the spinal cord, there are two parts that would have slightly wider diameter. The cervical enlargement and the lumbar enlargement. The reason those are enlarged because there are more neurons there. We're talking about up here, the neurons on the upper limb. Down here, the neurons that would be going into the lower limb. Okay. So anytime you see the cross-section structure of the spinal cord, and especially if you do not see the bone, two items to look at so you can readily figure out the position. The anterior or ventral median fissure would actually have a somewhat wider separation here. The posterior median sulcus is just something like that. Okay. So once you see that, you can readily figure out what's anterior, what's posterior. Some of the things to allow you to figure out when you see the gray matter, the anterior portions, the anterior or ventral horns is quite a bit further away from the edge of the spinal cord. The dorsal horn or posterior horns goes all the way to the edge. Just by seeing those allows you to figure out what's anterior, what's posterior. I think I have some pictures of the actual ones. Let's see. So you're looking at these things on the microscope slides. That's what you readily figure out. Okay. So yeah. Anterior median fissure, posterior. These are the posterior horns. The anterior horns are here. Gray commissure in the middle of that central canal. Okay. In the 5-unit course, this is one of the quizzes. I need the students to identify all these. Yeah, when you enlarge the spinal cord to this size, yes, then you can readily see the pia mater. Okay, so what's wrapped around the spinal cord is the pia. Right. So any questions on all these terms here? Okay. So this is just so you know. The different sets of neurons and where they are in the spinal cord. So both viscerosensory, somatic sensory neurons, both of them are the pseudo-unipolar neurons, and all their cell bodies will be at the dorsal root ganglion or posterior root ganglion. So on this side would be the dendrite, on the other side continuing into the spinal cord. That would be the axon. At the posterior gray horn is where you have the cell bodies of the interneurons. Interneurons or association neurons, their cell bodies are there. Okay, this is where they would be linking with these sensory neurons. And the somatic or somatosensory neurons would be linking with the interneurons much closer to the posterior horn. Then a little bit deeper is where they will be linking with the visceral sensory neurons. At the lateral horns, you have cell bodies of visceral motor neurons there. At the anterior gray horn, cell bodies of the somatic motor, 2 terms, somatomotor, 1 term. Okay. Somatomotor neurons. Okay. So now we're moving into PNS, starting first with the cranial nerves. Okay. Yeah, all textbooks would do for human anatomy would only deal with these ones that are readily visible. Okay. Essentially, all of them are macroscopic, starting with CN1 all the way to CN12. So the way these are named has to do with start from the anterior as well as the highest, and then continue the numbers with posterior and lower. So, so I mentioned when we're looking at the skull on the cranial floor, that's the reason why CN1 is number 1. That's most anterior as well as the highest. Then after that you have CN2 optic. So I also need you to know the functions of these. Get to know whether they are sensory, motor, or mixed nerves. CN1 olfactory nerve is sensory. Okay. So this is the one that is going to stay on the anterior portion, at the underside of the frontal lobe. And once it gets to the cribriform plate of the ethmoid, you would have a little bit of an enlargement here. That would be the olfactory bulb. And then setting off from there with a whole series of the nerves that is going to refer to as the filaments. And after coming through the cribriform foramina, they essentially will be stopping at the highest part of the nasal cavity, and quite a bit deep to the mucosa up here. So that's the only part of your nasal cavity that's responsible for olfaction. And also, the mucosa here would not have as much of the mucus producing glands as the rest of the respiratory mucosa. And also the olfactory tract here is going to enter into the limbic system. Okay. CN2 optic nerve also purely sensory. So they will be coming from the eyeball. Between the eyeball and the optic chiasma, that's optic nerve. After that, optic tract. Optic tract is part of the CNS, okay? Optic nerve is PNS. Not all nerves would be crossing here, okay? But a good number of them would do that. And of course, they get all the way back to the occipital lobe. Yeah, this is where they would be linking to all those relay neurons in the thalamus. CN3 oculomotor. This one is motor. Okay. Yeah, the somatic fibers is going to come to 4 of those 6 extrinsic eye muscles that are responsible for moving your eyeball. So so this is the one that's going to be working inferior oblique, superior rectus, inferior rectus, medial rectus muscles. So those 4. Yeah. So CN3 and CN4 will be issuing out of the mesencephalon. Okay. And this is mesencephalon here. And after that, it's going to come through superior orbital fissure, entering the orbit. CN4 is also motor, and it's coming further from the back. Okay, so what you see here, number 3, and then number 4. Okay, it's in the back of the a little bit under. The structure back here, and that is the corporal cortegramina here. So near the underside of that is where CN4 would be issuing. CN4 will be working one of the extrinsic eye muscles. And yeah, superior oblique muscle. And the reason it's called trochlear is because the tendon passes through that annular the annular ligament that's sitting here. Cranial number 5 is a mixed nerve. We are just mentioning the entire item. Okay. But there are 3 divisions. So the first 2 divisions are sensory. It's the 3rd division that is mixed. Okay. So first division is the ophthalmic division. That's the one that would be coming through superior orbital fissure. Once it comes through, it's going to stay attached to the ceiling of the orbit, and it would have a branch that would be working the lacrimal gland. Okay. Yeah. And then it will continue to come out of the supraorbital foramen or supraorbital notch. And then the branches would be going up the forehead and your scalp. So it picks up all the sensations of that part of your head. Okay. Totally sensory. the maxillary division is the one that is going to be going through the foramen rotundum. After going through the foramen rotundum, it's going to be passing across the inferior orbital fissure, and then it's going to stay on the floor of the orbit. And that's where you have the maxilla. Okay. And then eventually it's going to be coming through the infraorbital foramen. And yeah, once it enters the inferior orbital fissure, it'll be known as the maxillary nerve. And then once it comes out of the infraorbital foramen, it'll be known as the infraorbital nerve. And then all those branches would be picking up on the sensations of that part of your head. There are also a whole series of branches going to all your teeth in the maxilla. Those are the superior alveolar nerves, picking up all the sensations of the teeth on one side of the maxilla. And then for the mandibular division, that's the one that would be coming through foraminal valley. Once it comes through that, it'll be known as the mandibular nerve. And then it's going to enter the mandibular foramen. Once it got in there, it'll be known as the inferior alveolar nerve. And it's going to have branches that's going to be picking up all the sensations of your mandibular teeth on one side. And eventually it exits the mental foramen. Then it'll be known as the mental nerve. So it picks up on all the sensation of that part of your head. And then right about here, it will give off, as soon as it comes through the foraminal valley, it would be giving off several branches. And one of them is the lingual nerve. Okay. And that's the one that's picking up the general sensory of your tongue. Okay, not the taste, but just this general sensory, not the specific sensory, special sensory. So, and then it would have branches that is going to go to these muscles, temporalis, masseter. Those are the two major ones that is going to be lifting up on your mandible. It also has branches to medial and lateral pterygoid muscles. Okay, so that's the reason this is the mixed part of the nerve, okay, of the trigeminal. So we take apart the divisions, then you should know the first two divisions are sensory, third division mixed. Okay. Okay, cranial nerve number, yeah. The cranial nerves 5, 6, and 7 would be issuing out of the pons. Okay. So Number 6, abducens, is a motor nerve. It's the one that's going to be working on the lateral rectus muscle. The lateral rectus muscle is the one that is going to be drawing your eye for you to look out the corner of your eye. Okay. That's why it's referred to as abducens. What's it abducts the eyeball. Number 7, facial nerve is a mixed nerve. Mostly it has motor functions. That's so this is the one that had come through the internal acoustic meatus, and it works its way through the it works its way after entering the internal acoustic meatus. It works away the petrous temporal. Okay. Petrous portion of the temporal bone. So after working its way through that, it will be coming out of that opening between the styloid process and the mastoid process of the temporal bone. That's the stylomastoid foramen. Once it's there, it would be branching off into these 5 branches. And it would have branches going to all these muscles that is responsible for your facial expression. Okay.
Professor, I have a question.
Yeah.
I'm not sure about this, but are these the nerves that are like responsible for brain freeze? Like, you know, when you drink something cold and then it hurts like between your eyes, is that like related to here, or is that another nerve?
Yeah, this is the one. This is the one that would have branches from the temporal nerve. Okay. The top one is the temporal nerve branching off from the CN7, and the branch would go to your the muscle around the eye. This is a mixed nerve because it has special sensory impulses from the taste buds. So the anterior 2/3 of the tongue, the taste there is going to be picked up by this nerve. Okay. And also it would have impulses from skin on the ear. Okay. Number 8, vestibulocochlear nerve is sensory. In old books, this one's referred to as the auditory nerve, but ever since they figured out that there were two parts of it that came together to form it, then from that point on, it'll be referred to as the vestibulocochlear nerve. The part of this one that is at the vestibule, that's the one that's going to be picking up all the sensations, circular canals in the inner ear. Okay, semicircular canals is the one that's responsible for your sensation of equilibrium. Okay. Those three items here, the semicircular items here, have a whole series of cells in them. You also have fluid in them. And those three are the ones that allow you to figure out in any of the positions between the erect posture, horizontal posture, or that anywhere between those two is going to be picked up from here. So, and then all those branches are going to be coming together here. And that's what is referred to as the vestibular nerve. The other part would have a whole series of branches in the cochlea. The cochlea also, you have fluid in that, and you also have a whole series of ciliated cells in there for picking up the vibrations of that fluid. Okay, that is picked up with the the movement malleus, incus, and stapes. Okay, so when stapes starts shaking here, it vibrates. It whatever frequency it's handling with the fluid that's in here. And the ciliated cells are the ones that's going to be picking that up and then send the impulses into all these. So this part would be the cochlear nerve. And then once they are together, they'll be referred to as the vestibular cochlear nerve. So this, the top nerve issuing from the medulla oblongata. So this is the nerve that's picking up on both of them, both items. Okay. And because this nerve is the one that is going through the petrous temporal, and relatively next to it, you have your internal carotid artery going through the petrous temporal also. So usually anytime when you pick up on some of that noise of the blood flow has to do with the blood pressure, the blood that is heading to your brain. Sometimes you can pick up on that. Number 9 is the glossopharyngeal nerve. This is the mixed nerve. The glossopharyngeal is the one that's going to be working many of these skeletal muscles at the pharynx. And this is the one that's going to be picking up the taste in the posterior third of the tongue. And also have all the sensations of everything from this part of the pharynx. Yeah, it works the stylopharyngeus muscle. Yeah, this is the one that allows you to raise up the soft palate to close the back of your nasal cavity when you swallow. Number 10, vagus nerve, is a mixed nerve. And this is the only one that continues out, totally out of the head and neck region and moved into your thoracic, abdominal pelvic cavities, and would have branches going to all your internal organs. Yeah, you have somatic motor fibers here, working some of those skeletal muscles in the pharynx as well as the larynx. Yeah. The branch, it's sent out as a recurrent laryngeal nerve. Is the one that will be working those muscles, that will be working your focal fold. It also give you a whole series of these somatosensory impulses from tiny areas, the skin of the external ear. It's not the only one. You also get it a little bit from some of these others. Yeah. Number 9. Again, small area of the skin of the external ear. And then number 7, I think. Number 7 tiny patch of skin on the ear. So because you have a series of these muscles that would have sensations at that part of the of your ear, that is the reason when you stick something in your ear, you would go through the response by these the nerve functions of these nerve functions, coughing, when we get to the parasympathetic division of the ANS, we'll be going through a little bit more of what the vagus nerve will be doing. All right, number 11, accessory. And yeah, this is the motor nerve. It works your trapezius and sternocleidomastoid muscles. And this is the one that has a whole series of rootlets starting from C1 to C5. And then it shoots up through foramen magnum. And then it swing back out of the jugular foramen together with 9 and 10. Okay. And this is something that was noticed maybe a few decades ago. But because they already started with all these numbers, when they finally notice that this one doesn't really come out of your brain, they simply stay with it. Okay. Yeah, they couldn't change the number, so they just stay with that. And that's also the reason I refer this part as a spinal accessory. Okay. And then hypoglossal is also motor. That's one that's going to be working the extrinsic muscles that would be attached to your tongue. Those are the ones that would be moving your tongue. And then you also have branches going into the intrinsic muscles of your tongue. Those are the ones responsible for changing the shape of your tongue. Okay. So, yeah, get to know these functions, right?
And when you say get to know the functions, you're just saying to understand each nerve and whether it's mixed sensory or motor.
Right. There will be questions on that in the exam.
Thanks for the tip.
test tomorrow is going to have a whole series of matching questions for you guys. So hopefully it doesn't take you as long, because when you're doing matching, you don't need to read as much. All right.
Kind of like the multiple choice ones on the bonus points, yeah.
Yeah. Now moving on to the spinal nerves. For the spinal nerves, I just need you to know something about the ones that belong to these sets of plexi. Okay. By plexus, we are talking about those paired spinal nerves issuing out, and then they start combining with other nerves, and then they form a solid piece of nerve going to some parts of the body before they branch off again. So for the cervical plexus, I need you to know the phrenic nerve. Okay. C3, 4, and 5, they combine to form the phrenic nerve. The phrenic nerve would continue and it would be staying attached to the cardiac sac. And it'll be stuck on the surface of the pericardial sac. And then it'll reach diaphragm. Be there to work the diaphragm. And then in many of these pictures in your book, they listed a bunch of things that's referred to as the ventral rami. And here's the definition of ventral rami. Okay. This term we use for the spinal nerves. They'll be carrying both sensory and motor fibers, and they would be innervating the muscles, joints, skin. Okay. So that's what they mean by ventral rami. So it's just saying that for the cervical plexus, these 5 nerves will be doing all these things. This one also tests my students quite a bit, because in lab we would fully expose the set of the structures right about here. So for the brachial plexus, the roots would be from C5 to T1. Okay. And so five roots, and they all come together to form three trunks, superior, middle, inferior trunks. And then they spread out and form 6 divisions, 3 anterior and 3 posterior. And then those divisions would continue to form these 3 cords. And in the lab, there would be all these other structures around them. And that's when I tell the students that they are named for their positions. These 3 cords are named for their positions around the axillary artery. So the lateral cord is the one lateral to that artery. Medial cord is medial to it. Posterior cord is in the back of it or deep to it. And then they continue and become the should be a set of I suppose they just refer to these as nerves here. So what's happening is this. The lateral cord continues out as the musculocutaneous nerve. The musculocutaneous nerve is the one that's going to be working 3 muscles here. Coracobrachialis, biceps brachii, brachialis. And you'll also have branches continuing all the way past the elbow, and have branches to the skin and all that. So that's the reason this is the item that's referred as a musculocutaneous nerve. Okay, works 3 muscles, also takes the sensations from your skin, your forearm. The medial cord is the one that would be continue medially and becomes the ulnar nerve. Ulnar nerve have branches that would be working your flexor carpi ulnaris and flexor digitorum profundus. And then here, the lateral and the medial cords would form a large nerve. That's the median nerve. median nerve is the one that's going to be working all the rest of the muscles on the anterior forearm. Okay. Posterior cord continues out as the radial nerve. Radial nerve is the one that's going to be working all the muscles in the posterior arm and forearm, all the extensors. It's going to be worked by the radial nerve. One item that does flexion is worked by the radial nerve because that muscle started off in the beginning as one of the posterior muscles and ended up laterally. And that would be the brachioradialis muscle. Okay. In the lab, the way once we have this part exposed, and that's what I tell the students to look for. Find the M. Once you find the M, you have musculocutaneous, median, ulnar nerves. Lumbar plexus. This is the one I need you to know. The first two, femoral nerve and the obturator nerve. Okay. Femoral nerve is the one that is going to be, well, these ones are coming from L1 to L4. And the femoral nerve is the one that is going to be working the anterior thigh muscles. Okay. Your sartorius, your rectus femoral, etc. The obturator nerve is the one that is going to be working all those muscles on the medial compartment of your thigh. All the adductors are worked by your obturator nerve. Sacral nerve, sacral plexus, L4 to S4. Main one I need you to know there is the sciatic. That's the largest nerve on you. And that one is formed by L4 to S3. And yeah, it's the biggest nerve on you. And that's the one that's going to be running almost everything. Of your lower limb. Okay. Go over these ones in 10 minutes. All right. The somatic reflex arcs, these are often referred to as simple. And you have the monosynaptic and the polysynaptic. And anytime when you need to set up a drawing of these, you need to label at least 5 items here. For the monosynaptic, sensory receptor, sensory neuron, synapse, motor neuron, and effector organ. If you do not label the synapse, you lose some points. If you title a drawing monosynaptic or polysynaptic, and you do not label the synapse,
So Instructor Jet, you want us to label it just as it's shown there. So you want us to write everything that we see in that picture, just the way it is.
the
Don't
reason
leave anything out.
the reason is that if you don't identify the synapse, nothing happens with no synapse. Okay? Nothing happens.
Okay.
So for the polysynaptic, again, sensory receptor, sensory neuron, and synapse there for the interneuron, and another synapse for the motor neuron, and then the effector. So just before I forget, in the exams on the filled-in portion, and you've probably have gone through that quite a bit with the PAL quizzes, especially eventually when you need to fill in something. When you misspell that item, it's going to be marked wrong. And the way I am by someone who keeps misspelling everything when they fill in for that, for the first 20 questions in the exam. It's convincing me that the person doesn't have a high level of intelligence. Many of those items are in the exam questions. There's no reason for misspelling them.
Actually, though, it's really hard when you have 45 seconds to spell a complex word, and we're already nervous. So we're doing our best. We have high intelligence. I mean, some of us have
There
made the
is-
dean's list twice. So thank you.
All those items, almost all of them, are in the questions. You cannot go back to change it. Tough.
In that regard, Professor, I'm wondering, how do you want us to spell gray matter? Because the way you spell it is Grey. In our text, it's Gray, and in other resources, it is Gray. As a journalist, Grey is how British spell it. In the US, it's Gray. So I just kind of want you to clarify, how do you want us to spell gray matter?
That's fine. You can also pronounce maxilla, capillary. Yeah. Autonomic nervous system. Anytime you see this term, associate that with visceral material. We're talking about handling internal organs, and that these are things that most of the time, almost all times, you cannot consciously control. The two subdivisions, sympathetic division, that's the division that is going to be how you respond to emergency situations, fight, fright, flight. And the set of the cell bodies here for the sympathetic division is from T1 to L2. T1 to L2. Okay. Those are the branches going to all these internal organs. Parasympathetic division, cell bodies will be in the nuclei of cranial nerve 3, 7, 9, and 10. And then S2, 3, and 4. So all these ones up here would have the cell bodies at cranial nerves 3, 7, 9, and 10. And then these ones for the working on the bladder, urinary bladder, and the genitalia, S2, 3, and 4. And parasympathetic division goes on when you're resting and digesting. For the somatic efferent and the autonomic nervous systems, it's quite a bit of difference between the somatic motor. But then for the afferent portion, it's quite similar. So for the sensory pathways, in the ANS, would be very similar to the somatic sensory. So you saw that before. Both have the same type of neurons, sensory neurons, and the cell bodies are all at the dorsal root ganglia. So now, so this is essentially going over on some of the differences between them. Okay. And then two other terms to note. Also, once you know these two terms, it also direct you to where their cell bodies or roots are. Okay. Parasympathetic division is often referred to as craniosacral, simply because of CN 3, 7, 9, 10, sacral 2, 3, and 4. Thoracolumbar, T1 to L2, that's sympathetic division. So once you know these, then you can figure out which one is doing what as we continue here. Starting with sympathetic division. So once you know where the cell bodies are, the major function is the one that is going to give you strong action during emergency situations. Once you pick up stress, fear, rage, pain, and some serious injury, the sympathetic division is the one that's going to be acting globally. And it's done with these two hormones, epinephrine and norepinephrine. And most of these two hormones would be sent out from the medullary region of the suprarenal glands. Suprarenal glands are the ones that is those almost pyramid-shaped organs sitting on top of your kidneys. And most of the time, I just stay with suprarenal. Your book still stay with adrenal. Okay. The reason scientists are staying with suprarenal, so because some drug company patented adrenaline. So that is the reason we do not use adrenaline here. Epinephrine instead. And scientists use suprarenal as well as epinephrine or epinephrine because they don't want to sound like sport sports reporters okay, saying that the athlete is going through adrenaline. So, and the reason it acts globally with these ones is that these ones being sent out by suprarenal gland, it goes directly into the blood circulation throughout the body. So all parts of the body will be exposed to that. So some of the major actions here: peripheral vasoconstriction, and that happens by blocking most of the blood that would be entering into the digestive tract, as well as the areolar connective tissues. And that's how you raise your blood pressure. You'll also be increasing the heart rate as well as the output. You have a much stronger contractility of your heart muscles. Dilates the pupil. And that's definitely has to do with whenever something jumps out in front of you, or you're walking out around dusk, something jump out of the shadows in front of you, immediately your pupil need to dilate, the joy and better focus on what you are seeing in front of you. Also increase glucose consumption. Sympathetic system also be responsible for the ejaculation as well as the contraction of the vagina. The sympathetic division would have short preganglionic fibers at the origins portions at T1 to L2 or 3, and the synapse with the posterior ganglionic neurons. After the synapse, then those are very long fibers. So short and then long. What do we mean by that? Cell bodies of these neurons are in the lateral gray horn. From there, coming to the sympathetic trunk ganglion. This is where they would be synapsing with the next neuron that's going to go from here all the way to the internal organs. So from spinal cord to the vertebra, that's a very short distance. From the vertebra to the organ, that's much, much longer. So we deal with this one as short and long. Okay. So okay. Yeah, so these ganglia, this set of ganglia tend to go from T5 all the way to L2 or 3. Sometimes these are referred to as the sympathetic chain ganglia. Sympathetic again. Yeah. Okay. So the next one, parasympathetic. Okay. Actions. Okay. This one is going through in handling resting and digesting, and those functions has to do with your digestive organs. And the actions here is totally local. This one is not sending out anything that is going to be circulating in the blood. No hormones. Okay. So the major actions: vagus nerve, releasing acetylcholine on SA and AV nodes in the heart that will slow down the heart rate. You'll be increasing the digestive tract. It would cause the set of plexi in your intestines to work a little bit quicker, as well as getting those two plexi in the intestines to also work a little bit more on the gland, the secretions of the glands. This one is going to constrict the pupil when you're sleeping. You don't need to have your pupil drawing in more. Brightness. Parasympathetic division is the one responsible for erection. We're talking about sending blood into the erectile tissues at the penis as well as the clitoris. And
Professor, did you say it increases blood flow?
it just causes blood to send into wherever you have erectile tissues.
So then do those medications like Viagra and stuff interact with the nerve?
Probably. That's also why the med students remind themselves. Parasympathetic keeps it up, sympathetic shoots it out.
Gotcha,
So,
that's good to know. Thank you, Professor.
Convenient.
long. Now this one is going to have long pre-ganglionic fibers. And these are the origins, CN 3, 7, 9, 10, S2, 3, and 4. And from there, the neurons, the axons of the neurons go all the way to the organs. At the organs, they would be forming synapses with the next set, the postganglionic neurons there. And of course, those neurons would have a very, very short fibers because they're already inside the organs. Okay. Yeah, the parasympathetic ganglia in the wall of the organs, they're microscopic and also not named. So as you go through autonomic nervous system in the textbook, at best just stay with the first 2 or 3 pages. Don't go any further. Okay. Just stay with the terms I have here. Took a while before your textbook finally agree to mention a paragraph of this in the digestive system. I just need you to know that your digestive system has something that is referred to as intrinsic innervation. Okay, and this has been known for quite a long time in but textbooks only include that recently. This is the ENS, enteric nervous system. This is an intrinsic innervation. It has 100 million neurons, and it works on the motility as well as all the secretions of any of the cells that are producing material to send out, as well as all those multicellular glands in the digestive tract. In a healthy person, the activity is somewhat modulated by the sympathetic and parasympathetic systems, but the ENS is capable of functioning entirely on its own with these two plexi. The submucous plexus used to be called the Meissners. That is the one that continue into the submandibular, goes into the submucus as well as the mucus layer, submucosa, and the mucosa. And that's the one that's going to be working everything there. All the glands, and the two sets of smooth muscles, the muscularis mucosa. Okay. Myenteric used to be called the Auerbach's plexus. It's the one that stays at the muscularis externa. And that's the one that will be working. The muscles will be doing segmentation and peristalsis. The way the branches of all these two sets of nerves in your intestines is not like what is drawn here. It's more like how it would look if you wear two pairs of socks on each foot. And because this one is intrinsic and it's its own system, when someone is considered brain death. But if they can still work on the device that would be getting the heart and lungs work and send nutrients into the digestive tract, all the rest part of the body would still be regarded as alive. So the autonomic nervous system, get to know what is shown here. First synapse. For both of them, for both parasympathetic and sympathetic, neurotransmitter sent out acetylcholine. At the next terminal, second synapse, for the sympathetic division, that's going to be getting norepinephrine and then they also would be releasing acetylcholine onto the skeletal muscles. By cholinergic, we are talking about acetylcholine. Okay. By epinephrine, we are talking about something that's adrenergic. Adrenergic used to be adrenaline. That's why adrenergic. Parasympathetic division at the second synapse would also be releasing acetylcholine. So as we go into here, for the somatic motor, okay, talking about what we're dealing with when we're in the muscular system. So those myelinated cells would be sending out acetylcholine onto the skeletal muscles. But once we get to the autonomic nervous system, the sympathetic division at the ganglion, the first synapse, acetylcholine, second synapse at the organs, norepinephrine. At the same time, it would also be sending acetylcholine onto the medullary division, of the suprarenal gland. So the suprarenal gland will be sending out epinephrine and norepinephrine in the blood. So in addition to where the organs are getting norepinephrine, all parts of your body will be exposed to epinephrine and norepinephrine. The only exception is for the blood vessels at the skeletal muscles, because in the emergency situation and you need to start moving, the skeletal muscles would need as much blood as possible. So that's the reason for that. Now, for the parasympathetic division, at the first, it's again long and then short. Okay. So once it gets to the organ, you'll be releasing acetylcholine, and then the post ganglion axon would also be releasing acetylcholine. So that's how all that works. Yeah, that's all the material I want you to deal with for the ANS. Okay, just material mostly in just the first couple of pages in your in that chapter. Yay.
Professor, just out of curiosity, are you going to let us go earlier to study again?
Pretty much. I am done with the material on the exam. Okay. So I may start a little bit with the urinary system after the exam tomorrow. And I may also show you more videos from BBC. Okay. So I'll start closing now.
Thank you, Professor.
Good night.
Good night. Love you. I mean, sorry.
Oh.
Sorry.